So if you have questions, use the chat box so we can ask those questions or at the end or during the talk if the opportunity arises. And we have an interactive session for you today. So there will be some polling and some questions during the talk. So if you are ready, I think we have already 46 participants in the room waiting. Not for me, but waiting or for Alexander to start. Because this is the 10th IPPN webinar. That's already quite something. So I'm in the imaging working group. So it's part of the International Plant Phenotyping Network. Today we have Alexander Bucksch with us from the University of Georgia. And I'm Rick van der Zedde. I work in Wageningen in the Netherlands. And this is also actually my last activity that I do from the imaging working group, because I have been selected in the board of IPPN. So I will still be very involved in this cool network. So the imaging working group consists of Tony Pridmore from Nottingham, Sotosh from Edinburgh, Hanno Shah from Jülich, Scott Chapman from Australia, and myself. And maybe you've noticed, potentially we also have some new people, but we have recorded all those webinars, and you can go to the IPPN website and you can watch all these recorded webinars from really excellent speakers. So there's a lot of inspiration there if you are in a lockdown, and the whole world is actually facing some kind of a lockdown situation. So. But let's move on. I would like to introduce you to Alexander Bucksch, University of Georgia, and he's got a lot of stuff to share with us today. So I will hand over the word to him, and then he will share his screen, and I will mute myself and also will listen to what you have to say. So Alexander, if you are ready.
I'm ready, yeah. Thanks for the introduction. So let me share my screen quickly. And you should be able to see that now.
Yeah, works.
Is it working? Okay. So yeah, as Rick said, so I'm Alex Bucksch. I'm from the University of Georgia, and today I talk a more extensive version of the talk I gave already at the Genomes to Field workshop at Phenom this year. So it's all about how can we make new methods that use a lot of data out there to find or describe uncharacterized phenomena. So phenomena that we might not even know about. And for those who do not know that, my background is actually in computer science. And that's why I want to talk about how we use basically massive computation for roots, and especially big data of roots that we collected in the field. So I guess there are a couple of computer scientists also online because it's an imaging webinar. So the basic motivation for anybody who wants to go towards plants in there that comes with a very technical background is simply to recognize, okay, food sources, carbon sequestration, all of that feeds into the basic layer of the human population. So if you don't fulfill your physiological needs, none of the of the other needs of humans are actually existing. So you need plants for making shelter, like houses, or generate energy, simply for everything. I think that's a good motivation to go towards plant biology as a computer scientist or mathematician, just in general. So the next question that you might have if you come with a more technical perspective is how do you link these technical aspects into a biological context, or how does it become useful? And there are basically three areas that my lab works on or is involved in. And one is, yeah, we compute on very large and complex data sets, so we are not afraid of using gigabytes and terabytes of data. Then we use our technical algorithmic skills to detail phenotype to a level that is not doable by hand. And then sure, what I'm talking today about is really, can we actually find unknown or uncharacterized phenotypic patterns that are hidden in the data that is already collected, or can we collect extended data sets to find these new patterns? So how does this look like? Just from a very abstract point of view and high level, there, also like a data descriptor, or in what we call computational plant science, one big pipeline, and that is a description. If you have imaging data or any kind of sensor, so we take then this data description and start analyzing it to see, is there any kind of regularity in it, or is there a pattern that we didn't look at before? You can do the same thing with having a mathematical model that generates then such a data description, and then you start again analyzing the data description that you generated. That's just a high-level overview of what we're actually doing, and it brings me into the question that I really want to answer or show as a problem here in this talk. That is simply, can we discover unknown phenotypic patterns in this big data of roots that we collected over the years? So a lot of that is actually done together with Momo or Limeng Xi that is working in my lab, and she did a lot of the computation that I will show in the next 30 minutes, I guess. So, all right, let's go back to the very basic idea. If we look at roots, and if you just excavate roots in the field, you will see there are phenotypes. They are related to, or they change with the environment. So for example, if you look at the upper root here, that is a common bean root that is grown under drought conditions, and the same genotype is grown under well-watered conditions, and you see things changing. So basically the angle, how shallow the root goes into the soil, or also you see on the well-watered root that there's more adventitious root growth than on the drought root. And normally we think, if we can describe or quantify numbers, how these changes differ, significantly, we must be able to breed these traits or these descriptions of the root. But if you look at variation between environments, within environments, between genotypes, whatever, there's the first big question that we actually have. That is, how do we really capture variation? So we collect all of these data all over the last years. And then the question is, do we actually capture all of that variation? And I just wanted to have this little poll here to see, how does the community actually capture variation? And what do they mean by variation? So the idea is, over the last perhaps 8-9 years, all over in 3 different continents, my group, or previously myself with collaborators, we were simply collecting a lot of root data, and we got in contact with a lot of protocols. And I give it another 20 seconds probably for everybody to vote. We have already half of the users voting. Let's go with this. I share quickly the result and who is the winner in this very quick poll. I hope everybody can see that. But a fairly large number actually says we just look at what is a representative sample, or we just take random samples out of the field, right? Not many say, okay, 1 or 2 samples are enough. And another protocol that makes me happy is actually one person said there's something else. What I want to point out here is a representative sample that is often used, and over 40% of the voters just did that, or said they do it that way, is definitely insufficient. Because if you look at bean roots, that's an example of a bean root or common bean root that I often get that's the representative root structure that we should look. Now the reality for this genotype is this. We see actually many more architectures, not only the one that I just showed you that is the representative ones. We see actually many more architectures in one genotype, and these are kind of neighbors in the field that you see in this slide. So the solution that we had is simply, okay, let's take all of them out and we look at 5 to 1,000 individuals that are grown in the field next to each other. And for the rest of this talk, I will focus only on this one genotype that we had in 2 environments, in water stress and non-limiting conditions, which is the L8857. And we have grown them in Wilcox, Arizona at the Root Biology Research Center in 2015 and 16. So there comes the second big question. So how do traits or measurements that we take on roots actually capture the shape of the root system? And there should be a second poll now somewhere. So how well do we describe the root system of a root or of a plant when we just measure the way we do? So we have now either in your protocol, you use a representative sample or you take just random samples, but then you start describing a root system. So in this poll that I just started, I just want to know, how do you think you describe or how well do you describe the root system shape? And I just go on talking and keep this question on for quite a while now, for about 2 or 3 minutes. So what most folks do that I met out in the fields is they start measuring something like width or diameter or angle at a certain location within the root system. Like if this is a root system that you see here, we would just take the width here at this defined location, which is perhaps 8 or 10 centimeters from the first basal root upwards or something like this. So that's where the root system width would be perhaps defined, or the angle or something, some other trait. You do that for all the roots, and then you know already, hey, there's a lot of variation between root architectures going on here, and then we get 3 measurements. And if you do it more and more and more, and we end up with something that is a normal distribution, simply by measuring all of that variation that is out there, either on representative samples, or if you just take random samples out of the field. It wouldn't matter in that case. It's just a little difference in the distribution. And we can ask the question, does this now describe anything about the complete root system or about the complete architecture? And my answer is typically no. Because if I start measuring the same roots at a different location in the root system, and I just keep on going with this example of the root system width, and I just look at a little bit lower in the root system, this would happen, right? We would get another measurement, and now exactly with this root here, you see something happening where you just see that the rank, the order of these points is actually changing. But still, we would get a second normal distribution looking a little bit different if you do that on many, many, many different roots. So what I want to point out here is simply that whenever we start measuring just at one location within a root system, we never know which architectures we are actually looking at, because we don't know how the individual measurement points that we have taken are actually connected to each other and how the complete root architecture is described by it. We are just looking at localized trait measurements. And if we want to look now at this poll, and we just share the results, we see maybe if we just measure the same trait at many locations in the root system. So yes, that's right. If we get indefinitely many measurements throughout the root system, that could be good. But still, larger numbers that here traits were not meant to describe complete root architectures. I think there's some truth to that too. If you just want to look at diameters within a root system, there's definitely some truth to it that this was not necessarily meant to reconstruct a complete root system just from this one trait. And there are other still to about 20% that traits never capture variation in the root system. That's also kind of an opinion that you can take. It all depends on what do you define as trait. And I think it's still a long discussion to define what is meant by a trait. Definitely, if you just measure at one location, you don't know how these are connected. If you measure at just two locations, you don't know what is happening in between, and you need many, many more measurements. So let's look at this, this picture for a second. So what we did is taking really, really a lot of measurements on individual root systems that vary by architecture. And what we do is we're taking the root system width, like we just did in this little example, but we take it on both sides and get then an average line here. That is this red line in this red root system here on the left. And what we take from there is we take the accumulated width and just draw it as a function. So that is the middle red line that you see here, and the red line at the root system is simply the average between both outlines. So that is somehow giving you an idea of how much width is accumulated at a certain depth. And as depth, we just look at what we can excavate with a normal shovel in the field, following the shovelomics protocol. So this curve reacts quite nicely to the root architectures that we see in the field. And that's what we want to use throughout the rest of this talk. So if you do that on a lot of roots, like we did at the Root Biology Center there, on about 500-600 roots, you end up with a big, big mess. So if you look at the right, that's just a water stress study, and these are all the curves we extracted. So one thing to mention here is these curves are drawn in a different way than on the left. So what this is representing is basically the change of the D curve here on the left side. We are just looking here at the changes within these curves. This is just easier and numerically to distinguish down the line. But that's the only trick that it's happening, and that's why it forms these arcs in here. Still, you have here the excavation depth on the X. Just up here, we are looking at the angle to the X just to encode it a bit nicer. But yeah, it's a big mess. And if we look very clearly, there's no difference between well-watered and water stress conditions, right? We just get 2 times the mass back where we cannot infer any information that we would normally see if you just say, okay, this is representative sample A and B, and then often you see differences between these environments. So that would not happen if you just look at all the roots and just collect them all in the field. So we know all of these are in there, right? And they are very different. So what we want to go for is, we want to see, can we group all of these curves into very different architectures? Just like we had in this plot before, where we said, okay, there must be different architecture types if we just know how they are connected, which we can do with these kind of curves. So can we get to different clusters of curves? And that's what we did with this algorithm here. So what we use is the platform DIRT that I developed a couple of years ago, and we get these DS curves, which captures more or less the whole root architecture. So, or let's say parts of the root architecture. It actually misses out on the density. I want to be honest about that. But overall, the shape is pretty nicely described. So let's say through the outline, and we use a k-means clustering approach that that we apply to these curves to minimize the inertia around the mean curve. So what you see here is they're like different kind of clusters coming out that are very characteristic and very, very distinguishing between the root architectures. And then there's another part to it, because we are using a k-means clustering. We don't know what users are in there. We have to determine how many clusters are actually in this whole group of curves. So we have to estimate that or find out how many clusters we want to have as an input to k-means. And here I want to go now this time a little bit more technical, simply because quite a few folks at Phenom asked about how do you actually find out the number of clusters as an input to a k-means clustering, since many people apparently use k-means in other applications. So that's our way. So what we do is we basically run k-means 1,000 times, and that's where it becomes really computation very heavy, this algorithm, 1,000 times, and assume simply in the first step that we have 1 cluster to 25 clusters. So we have, for each of these assumed clusters, we run the algorithm 1,000 times. That's what we do. And then we use an algorithm called k-needle that we use to determine how many clusters are in there. So if you look at the right side, there are curves that that my student Momo did. So you see we run it for all the 25 clusters, and we see the inertia going down, right? And then we connect basically the first and the last point of this curve and use the distances to the curve, what is happening in this figure B, to determine a difference curve down here in figure C that has an E point, right? So there's a normalization procedure going on that they always look down, and that's why the curve flips from A to B. And then we get this knee point in the difference curve here, up here, which points in the figure D then directly to 5 clusters. So these 5 clusters are what is describing most of the variation that we see in our curves, in this big mass of curves that we have. In the very last step, we take this number k, which is 5 in the example, and just run the k-means 1,000 times again. And we use the result that has the minimal inertia from these 1,000 runs. Why do we have to run it again? It's very simple, because every k-means algorithm uses a random seed as a beginning. So we want to overcome this influence of that is getting into our results just because of a random seed problem. All right. Let's get the second very technical slide before it gets easier again. The second question that I was asked a lot during Phenom after the talk there is, how do we really deal with these outliers that are still in the clusters? Because there are definitely outliers in these curves that are just resulting from the shovel digging into the root here and there, where you just have no control over. And there are outliers you just deform the root by means of this, of forces basically. So there are two ways to think about it. So one is that you have a magnitude outlier, and the other one is a shape outlier. And if you just want to talk casually about it, it is a shape outlier is simply a curve that does not really follow the obvious trends in the cluster. So it wiggles around the mean curve. If you look here on the left side, you see it goes a little bit with the trend, and then pew, it goes all the way down here and shoots a little out, and then the same happens for this example for this green curve here. And the other one is the magnitude outlier, where we just say, okay, this peaks out of the typical bandwidth. So what is the bandwidth? The bandwidth is what you see here on the right, where we say we have an envelope curve on the top and the bottom of the cluster, and then we have the mean curve, and we go to 50%. We cover just 50% of the to the left and the right of the mean curve, or up and down of the mean curve. So that's simply what we define from literature, and down here is actually the nice paper that we found. That's what we define as the magnitude outlier. So everything that peaks out here, we can more or less ignore. If you look at our two curves, or the curves in this cluster, we can write that in an outlier graph or an outlier plot, where the upper dashed line simply gives you a maximum that you can never overshoot. So that is just given by the space. That's the form of the space. And then you have a lower dashed line that is coming from literature, which we talked from this biostatistics paper, about 15 to 18% or a fraction of 0.5 to 0.1, 0.15 to 0.18, and what is going until 0.85. So all of that is called the variation in the curve, and defines more or less outliers if you're out in this space down here, where you see the greenish and purplish point. I think that's right, because my monitor just turned off here. Okay, it's back. Okay, so what I just called magnitude outliers and shape outliers are actually called in literature then modified bandwidth, which is the magnitude outlier, and the modified epigraph index, which is the shape outlier. And we are looking at this space that is kind of bell-shaped. And then we see in this inner space below the second dashed curve, which is an outlier or not. And that worked for us very well. Okay. We termed this, the result of all of these clusters, a phenotypic spectrum, because we have a large amount of architectures that we wanted to group. And that is the result we really see. So that's not a quantitative result. That's not anymore an idea for the genotype that I promised to use here in this presentation, which is L8857. And we see that in different fractions, we can cluster the same curves in both environments, in water stress and in non-limiting environments. And the question we ask there is, how stable is this? How much does this actually reoccur? And we calculated the stability, which is simply the fraction of curves that is never assigned to another cluster than the one it is in right now. So basically the stability says here in the first water stress environment that 66% of the curves have never been assigned to another cluster. Even so, there's a random selection or a random seed in the algorithm before. And these are quite high numbers, to be honest. So that you normally don't see that high. So that gave us a lot of confidence. And we see that, for example, the third architecture here, or the third shape, is has 29 in water stress environments. And then we see that suddenly the dominant and representative sample that is often used becomes, in non-limiting environment, becomes the second architecture, the fourth architecture, if you look just at the fractions. And that makes somehow sense to us. So why does the representative sample shows differences often, while the collection of all the samples does not show differences? That said, that happens also in the second year in 2016. Just the fraction overall is a little bit different. And at the moment, we think that has to do with the global weather pattern that was in Wilcox in Arizona. But we see that the differences between the fractions are pretty much the same than in 2015. And that is that the stability of the clusters is very high, just as before. Okay. How can we compare these 2 years to each other and these environments to each other? So there's another trick from shape theory that we use, and that is the Fréchette distance. So the Fréchette distance is very nice. It's a very nice tool to match up curves to each other. And what we do is we just measure basically the distance if you walk on both curves, and then have a distance between them, and then we sum up this distance. And that is called the Fréchette distance. And then you see that if you do that all versus all, so if you compare all the mean curves to each other, the following happens. We see here this matrix. We see quite a few clusters arising. And actually we see arising 6 clusters, which would say, okay, there must be 6 architectures somehow, because we cannot match every architecture to every cluster. And that's right, what happens here. Oh my God, who is writing this? So what happens here is that we match all these curves to each other, and we see that in 2015, 16, under well-watered and water stress conditions, the same curves are arising, so they are the same, and it exists across years and environments. So these architecture types, and we see here also an example that we pulled out of these curves that we have here, and it really looks quite nice, and you see the differences between the architectures arising. One issue we had is we had in the last architecture type that we determined with these clusters, we had a problem. We couldn't really match them nicely to each other using the Fréchette distance. What is here is that that we are unsure if we should make them now one or not, which is an open discussion that we just have with ourselves in the group at the moment. That is kind of an open question, because they're still very similar in how they just look like visually. But for the rest, all of these curves and these architectures really matched up very nicely, and they're all matched up with very high confidence here. So you see that here on the left side where the distances become very, very small. Okay. There's one more question that we wanted to look at, and that is what happens actually above ground. And it's just a crude measurement, but we measure the biomass above ground. The biomass above ground turned out, even so there's all this variation between these architectures that we can clearly distinguish between environments and years, we can, oh, what is this SMD? So we didn't see much variation between the architecture types above ground. So it looked pretty much the same. If you look at the distributions between the years in well-watered conditions here, and it looks pretty much the same. And the same happened for water stress in the same year. It looks pretty much the same. So we didn't find any significant differences in the shoot biomass that is related to the difference in the architecture types. Okay, this is all field data that we took in Wilcox. So how do we go from here? And that is a bigger question, how to get more control over all of that. And there the phenotyping becomes very important, because what we do is, hard, oh, how nice. We build a big mesocosm now that is just where we can control every little, little sprayer in this mesocosm box and measure at 2 different depths the water content. So we measure at 6 inches of depth and 15 inches of depth. We measure the soil water content, and we have a computer program that regulates all these sprayers. So in that way, we can keep that relatively nice and regular, such that we can start looking at the root architectures in a more controlled environment, where we have also control about over temperature and other things. Okay. What you see behind all these nice scribbles of somebody that just has a lot of artistic energy, we are actually growing our first experiment right now, and it grows nicely. It looks nice and green. So if this person that draws here perhaps also uses green to do that, that would be nice. Bees, exactly. Bees should be in there too. We need pollinators. So no, please make green bees nice. Just make green bees now, because these the bean experiment are replicated in the greenhouse, and it grows, and we are pretty excited what is coming out of that. Okay, bees, there we go. So let's go back to the data that we can use, the data that I described, or the descriptors we described. So I mentioned the platform that we developed in 2014, and that is DIRT. And DIRT allows us to extract all of these D curves and the DS curves automatically. So that's where we upload all our data, and that is freely available. Currently, we have about 650 users worldwide and 40 new users per month. And if you look at the distribution, the majority of users is still from North America, but still in Europe, we have about 15% of the users, Asia the same, and South America, Africa is quite rising a little bit. The benefit here is there's a very simple and reproducible protocol that is everywhere in an easy way to build, and that just gives us the data that we looked at now. I want to give a little heads up, and for those that want to have a preview, so we are working on the successor of DIRT, which was standing for Digital Imaging of Root Traits. And if you go to this Portnoy link at Cyverse, you will find a preview of what is coming up, which will actually change the picture a little bit, because what you can do in DIRT will be just one pipeline in this newer, bigger platform that uses about almost every supercomputer you have a login to, you could use with that platform. So another preview we have is this very cool 3D roots that we can analyze there. These developments. Okay, this is creative energy that is just nonsensical. So sorry, you were better before, dude. So this Plant IT platform will also host a 3D analysis. And these, for example, for roots that we scanned with our own scanner here on the left, that just has 10 industry cameras, and this is mostly made with by Sujing. So we can use these 3D roots and just go into the complete root system and make every individual root in the root system we can start measuring. All right, that's more or less all I had to say, and I just want to have two things that you can take home. So what data can we use? So we use just now these whole root descriptors that they're already available in DIRT for our experiment that we just redid in Arizona with all these thousands of roots. And then I want you to take home that root architecture types exist, and they are existing across years and environments per genotype, and they are very reproducible. Just the fraction, how often they occur, is actually changing. So what is left for me is to say thank you to the funders, which is ARPA-E for all the technology that we developed, and the NSF Career Award for looking into this phenotypic spectrum, the university for the startup package, Kuro for paying the undergrads that patiently work on these things with me, and Exceed for the computing power we get mostly in Texas at the Advanced Computing Center. And definitely say hi to all the collaborators. And I want to point out here, Andries Terme, he's searching a job. He's very good. He's also ecophysiologist and looks at root systems, uses DIRT a lot. So if you have one, contact him. And then thank you to the whole lab. That's the current lab, everybody that is in it. And now it's time for questions because my time is up. Thank you.
Yeah, by several people, I guess.
No, it was actually just me and Jimmy. Beans are a very thankful crop. You can get them out very easily. It's not like maize. Maize is hard, or for example, wheat is also pretty hard to get out of the ground because it deforms a lot. these ones were easy for another reason too. So when we did these experiments, we had a couple of trials before where we actually selected also genotypes that are very stiff, right? So we knew that we could hardly destroy them, so we could be pretty rough in what we get out of the ground.
Okay, but there's a question waiting for you on
I don't see any
data recording method, but let's go. I propose we go down from the first question to the last question. We had quite some questions, so it will take us some time.
Okay.
But first question is simple. Chris Peterson asked around the fourth slide, I think it said one genotype, but then it says 3 genotypes, and then which is it? A very targeted question, actually, but just to clear his ideas.
Oh, wait. So which is it?
Slide number 4, I think.
Bang. It's this one. The red one, L88 57.
Yeah.
Okay.
So it says 500 times or 1,000 times one genotype in the grid, and then it says 3 genotypes. So which one did you do below the image?
We did all the 3 genotypes, but I just talked about the L88.
Yeah, so you planted 3 genotypes, but you analyzed only 1 genotype, right?
Oh, we analyzed actually all 3, but then I can talk forever. Then it would be like a 3-hour talk. So there are differences between it.
Yep. Okay. Let me see. Second question is, could we not do, like you could say, why do we need to extract features that might not necessarily mean anything? We could use a data-driven approach to extract a later space representative of visual features for root mapping to an intended class.
Again, what did
you could say more the data approach. So could we use a data-driven approach to extract a latent space representative?
I don't think you can get the same thing with a latent space. I don't think so.
So I think the general question is your data analysis approach very targeted on your data, or could it also be applied to different types of data?
It could be applied to different kinds of data, sure, but you need to describe the data yourself. So it's a question of how you model the descriptor of your data. So we use here now these D curves and the S curves to describe our data. But yeah, if you have other data, then you need other descriptors. So we just target towards the root architecture, right? And we summarize it into one function, in fact, that we start clustering. I do think this would be possible with, if you model a function that describes your data, yeah, then you can go with the same approach, right? You start clustering curves at the very end. It's nothing else.
Okay. Do you know of other researchers that have used your approach on shoot traits?
My approach, no.
Okay.
I think this is the first time I was actually talking about it.
Oh, cool. Okay. So the next question is from Chris Peterson. This is a bit on the way that you take those images and you start analyzing your traits. Look, isn't the width measurement, as you show it, totally dependent on how you laid out the roots on the table?
Okay, that was what I mentioned before. Yeah. The trick with these genotypes is, which took us a while, is that they are very stiff, and if you take them out of the ground, they are not really deforming much. So there's not much you can do to lay them out. They just basically fall into the shape. So we had in 2014, our first experiment, we had exactly that problem that we had genotypes that were just not stiff enough.
Okay, so that is yeah, I can understand.
Yeah, it's just handled by the genotypes that we use, and that's how we got around it.
Yeah, because his comment is surely these root systems will be narrower if you were hanging the root systems from the stem, because they are not at all self-supporting, but in your case, they are self-supporting, you could say. So for a different crop, it could be useless.
It could be useless. I would not do wheat with it.
Yeah.
Or you could. I couldn't do, for example, a full diversity panel with it. You're really looking within one genotype and the variation in there.
Yeah. Cool. Another question. Have you considered the Kalinski-Harabas criterion to estimate the number of clusters?
No.
Okay.
Very easy. Never heard of it.
No, me neither. So I cannot help you there. Okay. And then we got our artist painting on the on the wall. So there's a question from somebody who said, can you share the webinar online? And that's the answer is yes, because we will put it on the IPPN website.
Yeah.
And then there's another question. I think it's more on the recording method. So how do you extract data from three-dimensional root images, and how do you analyze data? I think you should explain the recording method actually.
Basically, it's a structure from motion approach where we just use, you just go all around the root. You can just, yeah, there was a lot of painting involved. So what you see here is simply there are 10 cameras that go all around this root system here and collect about 2,500, 3,000 images. And then we use structure from motion approach just straight out of the box, and then you get a 3D image out of it. So what happened is we used the visual, there's a visual structure from motion package, and we started actually modifying it.
You modified the source code.
Yeah, we modified the source of the visual structure from motion package. And what we actually did is we modified the way how features are matched between images. That was the optimization we did in order to get that computable.
Mm. So how long does it take to analyze one series of images of one root system?
It depends a lot on the complexity of the root system, to say how fine it is. These maze roots, they can take like an hour or two.
No, that's acceptable, maybe.
Yeah, that's why we use supercomputers. I think I missed that to point out during the artist contributions, but we are actually using quite a lot of supercomputing power all around the US. So through Exceed, and also at the University of Georgia, we use own cores that we have just for computing our roots. And that's also what the DIRT and the Plant IT platform does, in fact. They just plug into supercomputing environments.
Cool. Okay. So, I'm not sure if there are no other questions. Last question. Do you have a high throughput method for collecting roots?
There is. That is a good thing. Okay. So first of all, yeah, it's called shovels, right? We are just digging things out, like you saw in this at the very beginning. And there is a robot in the make. So these algorithms that you see here now on the slide, these 3D algorithms are meant to go into robotic system that is currently developed at Penn State. That will be basically a tractor going into the field, pulling out one root after the other, and then put it into an imaging chamber where you have also cameras inside, and then you just collect the imaging data, send it out to a cloud storage service, and from there we go and start computing. So that should be an integrated pipeline at the end of 2021, 22.
Oh, but you really drag the plant from the soil, or do you dig it out somehow? Then?
We still dig it out. I don't think.
Okay.
I don't think there will be anywhere soon a way to image roots with this detail in soil below ground in the field.
Okay, so if you have a few more minutes, there are some questions coming in. How do you hold the roots in taking the 3D image? I think it's linked to this topic that we are talking about right now.
How we hold them?
Yeah.
It's just a Velcro zip that we put in the middle. You see it in the video. We just put in parallel. The maze roots are actually not deforming much if you pull them out of the ground. They are very, very stable. At least the root crown we see here. Still, whatever we take out here is destructive, right? We are cutting off a lot of roots when we dig them out. We are just looking at the root crown that still gives us a lot of trait information, but we are not taking all the the fine laterals or anything out of the ground.
Okay, and then another question: to which soil depth did you capture architecture? How deep did you dig?
Oh, let's say to which detail can you say it again?
To which depth did you do the digging in the field?
The digging. Okay, it's, I would say, like about 50 centimeters, I guess. That's about what the big shovel was.
Okay. I also recall that some plants have like tiny hair roots that go incredibly deep. But that's something that you then don't record.
Yeah, no, we are clearly destructive with what we do. That has never been a question.
Okay, 2 more questions and then we'll finish this session. Have you considered using an air spade to remove the soil rather than traditional shovels? Or would that be too destructive for structures such as root hairs?
Okay, root hairs is a different topic. We don't measure root hairs.
Good, okay.
For this, you need microscopes, right? Root hairs are just one cell.
Yeah.
But
the air spade? I'm not sure what an air spade
I heard, they use it in Iowa. I think Pat Schnabel's group is using the air method and it works for them. And I think as long as you take very rough measurements like we do, that should be fine.
Okay. And then what's the cost? How expensive is a 3D setup?
The one that you see here is about $12,000. And it's made by hand by ourselves. So there's a little cluster connected that is made out of Raspberry Pis. So in this little box on the side, there are 12 Raspberry Pis in it that are working as a computing cluster to capture the data or the imaging data.
Another question actually that triggers me too, actually. So I'm also involved in EU programs that enable access to research facilities. So we have some very advanced tools. Not everybody has access to it. But
yeah,
do you allow access to your lab to work with this 3D setup?
Sure.
And how is it funded?
If you've got travel funds to visit the lab, yeah, then you can come, basically. That's what happened before. I think Koji was there already. Also, students from Thailand were already there. They come most often on scholarships.
Oh, cool. Okay. Good answer. Yeah. If we are allowed to travel again. That's a minor remark in this age. Okay, there's another question, and then we're going to quit. But okay, last one. Any suggestions if you want to study root development throughout some timeline? How should we do the root sampling?
Okay, one way is you go to artificial environments. Just do it in pots, do it in gel. That is one way to deal with root development. Another way to deal with it is you look let's say you take different developmental stages, you just dig them out and then you compare the commonalities between the developmental stages. It's up to you how far you go into detail. So if you want to look at something like the phenotypic spectrum, where you have looked at many different architecture types, there you have the thing, it gets really a big experiment, right? You have to, for every developmental stage, you make like 500 to 1,000 plans and so on, right? That gets a big one.
Yeah, that's right. Yeah.
And then you start
And
comparing.
well, I said it was. Yeah, and I said it was the last question, but Massimiliano from Italy, and he was already here half an hour before we started the webinar, so he will, I think, get the credits for being very interested, and also he gets then the last question. Do you conduct your trials only in very sandy soil, or do you have even clay or loamy ones?
Actually, the ones that you saw are clay soils. They're from Arizona. It's a pretty clayish desert. And the mesocosm at the end, that is kind of a sandy soil that makes it easier for us.
Okay, and do you flood these soils before you try to extract roots?
Yeah, yes. Makes
cool.
it easier.
Okay, then, and if others have more questions, then I suggest to ask them by email or approach you in another way. So, Alexander, I would like to really thank you for this cool webinar. Very inspiring. Yeah, nice approach, nice hardware. So I wish you all a happy Easter. And hopefully we will meet each other live somewhere in the world as soon as we are allowed to travel again.
Yeah, hopefully. Thank you.
Yeah. Good. Cool. So thanks. Have a nice day, everybody.